Answering MCAT questions in under 60 seconds. A rare autosomal dominant condition is caused by a gain-of-function mutation in a gene encoding a cell surface receptor tyrosine kinase. In affected individuals, the mutated receptor is constitutively active, leading to abnormal tissue development despite normal levels of its ligand, which is the molecule that activates it. Which of the following best explains why individuals who are heterozygous for the mutation exhibit the disease phenotype? So if they're heterozygous, that means that in this case, one of their receptors is going to be, or rather, one of the genes that encodes the receptor is mutated and is going to produce these constitutively active tyrosine kinase receptors, whereas the other copy of the gene, right, is going to be wild type and produces normal receptors that are appropriately regulated, okay? So what would we expect to happen phenotypically? Like which of these genes, the mutated one or the wild type, is going to manifest in the person, okay? Like which phenotype, right? So let's just look at these. So A, the mutant allele is silenced, but the wild type receptor is insufficient for normal signaling, right? Well, there's no reason to believe that, right? So there's no reason, first of all, to believe that the mutant would be silenced, right? Because if the mutant was silenced, it wouldn't explain why they would have the disease phenotype, right? But then the wild type receptor being insufficient for normal signaling also wouldn't make sense because they're saying that the disease phenotype here is the phenotype that comes from overactivation of this receptor. So the wild type being insufficient seems like there would be less activation than more activation, right? So that wouldn't make sense. Okay. Now B, the presence of the mutant receptor causes loss of function in both copies of the receptor through a dominant negative mechanism, right? Well, this is a very weird answer choice. There's no reason to assume that that would happen. I mean, there's not enough information there. And then also, again, a gain of function mutation with a tyrosine kinase receptor like this, right? It sounds like you would be activating some sort of signaling pathway. So if the phenotype that we would expect would be something where it would be like there would be more, the function would be exacerbated rather than there would be a loss of function, okay? So that wouldn't make sense. And then C, a single copy of the mutant receptor is sufficient to drive abnormal signaling independent of ligand binding. And that sounds correct, okay? Because think about it. If you have a gain-of-function mutation that will basically constitutively activate a signaling pathway, like for example in cancer, a signaling pathway that causes growth, right? In that case, having just one copy of that mutation is enough because having a wild-type allele that encodes for a normal wild-type receptor will mean sure that you'll have some normal receptors that are regulated normally. However, the other receptors that are expressed from the mutant allele will be doing their thing. They'll be constitutively active. They'll be activating the pathways by themselves. And so consequentially, that specific allele is going to dominate, right? And so in this case, this is kind of like a proto-oncogene, you know, if you relate this to cancer in some way. It's a proto-oncogene because here, having just one mutation is enough to cause the disease, okay? So, and then if we look at this, D says the phenotype results only when both mutant alleles are present due to the cumulative receptor activity, right? Well, again, if that were the case, they would not be exhibiting the disease phenotype, right? They wouldn't, but they're specifying that they are. So that's wrong, right? And so C really remains our best answer, and it makes sense, right? Usually gain-of-function mutations like this are going to really only require one allele to be mutated.